Autophagy, often described as the body's way of cleaning out damaged cells and regenerating newer, healthier ones, has become a focal point in the study of fasting and longevity. At its core, autophagy is a process by which cells break down and remove dysfunctional components, recycling them for energy or for building blocks to repair and renew cellular structures. Over recent years, researchers have sought to understand how fasting triggers this process, what duration of fasting is necessary to induce it, and how these effects differ across species, particularly between rodents and humans. To begin unraveling the connection between fasting and autophagy, it is crucial to appreciate the biological significance of autophagy itself. Under normal, nutrient-rich conditions, the process operates at a baseline level, maintaining cellular health and homeostasis. However, when the body experiences nutrient deprivation, such as during periods of fasting, autophagy is upregulated. This adaptive response helps the organism survive by providing an internal source of nutrients and by clearing out potentially harmful cellular debris. Dr. Valter Longo, a prominent researcher in the field of fasting and longevity, has conducted groundbreaking studies on the effects of prolonged fasting. His research, often discussed and expanded upon by scientists like Guido Kromer, has illuminated key aspects of how fasting can trigger autophagy and even promote a phase of regeneration, following the breakdown of old or damaged cells. In rodent models, particularly mice, prolonged fasting of approximately 48 hours has been shown to strongly induce autophagy. This duration, while relatively short in absolute terms, represents a significant period for these small mammals. In studies, a 48-hour fast in mice not only triggers robust autophagic activity, but also initiates a phase of programmed cell death, or apoptosis, in certain cell populations. This is followed by a remarkable regeneration phase, during which stem cells proliferate and replace the lost or damaged cells, effectively rejuvenating tissues. The translation of findings from rodent studies to humans, however, is not straightforward. There are fundamental physiological differences between the two species, particularly in metabolism and body composition. Rodents, with their higher surface area to volume ratio and rapid metabolic rates, experience the effects of fasting much more rapidly than humans. For example, a 2-day fast in mice can result in a dramatic 20% loss of body weight, an amount that can be life-threatening. In contrast, humans typically lose only 1 to 2% of their body weight over a 4-day fast, a much more manageable and non-lethal figure. This discrepancy is crucial when considering how long humans must fast to achieve similar levels of autophagy induction as observed in rodent studies. Research into the circadian rhythms of autophagy, such as the work by Kate Thompson, has shown that in mice, the liver exhibits increased autophagic activity during the day when food is withheld. This suggests that even relatively short periods of fasting can have measurable effects on autophagy in certain tissues. However, the magnitude and systemic nature of the autophagic response appear to be much greater with prolonged fasting. Turning to human studies, the evidence suggests that a more extended period of fasting is necessary to induce significant autophagy. Investigations into the behavior of circulating white blood cells during fasting have provided valuable insights. These studies indicate that fasting for 3 to 4 days is required to observe a marked induction of autophagy in humans. This duration aligns with the metabolic differences noted earlier and reflects the slower pace at which humans deplete their nutrient reserves and initiate cellular recycling processes. The process unfolds in distinct stages. In the initial phase of fasting, the body exhausts its readily available glucose stores, primarily in the form of glycogen stored in the liver. As glycogen becomes depleted, typically within 24 hours, the body shifts to mobilizing fatty acids from adipose tissue, leading to the production of ketone bodies. This metabolic switch not only provides a new source of energy, but also serves as a signal to upregulate autophagy. The longer the fast continues, the more pronounced the autophagic response becomes, as the body increasingly relies on internal resources for sustenance. Interestingly, the induction of autophagy during prolonged fasting is not uniform across all tissues. Some organs, such as the liver, are particularly responsive, while others may require even longer periods of nutrient deprivation to activate autophagic pathways. The interplay between circadian rhythms and fasting further complicates the picture, as certain times of day may be more conducive to autophagy, depending on the organism's habitual feeding patterns. The regenerative phase observed in rodent studies following prolonged fasting is of particular interest. After a period of heightened autophagy and cell death, the body initiates a wave of cellular regeneration driven by the activation of stem cells. This phenomenon has been described as a reboot of the immune system and other tissues, offering tantalizing possibilities for therapeutic interventions in aging and disease. While direct evidence for this regenerative phase in humans is still emerging, the parallels observed in animal models suggest that similar mechanisms may be at play. It is important to emphasize that fasting, especially for extended periods, should be approached with caution. Individual responses to fasting can vary widely based on age, health status, and underlying medical conditions. While short-term fasting is generally safe for healthy adults, prolonged fasting can pose risks, including nutrient deficiencies, electrolyte imbalances, and exacerbation of pre-existing health issues. Medical supervision is recommended for anyone considering a fast of more than 24 hours, particularly if they have chronic health conditions. The scientific community continues to explore the optimal fasting protocols for inducing autophagy in humans. Intermittent fasting regimens, such as time-restricted eating or alternate-day fasting, have gained popularity for their potential health benefits and greater ease of adherence compared to prolonged fasting. While these approaches may induce autophagy to some extent, the evidence suggests that the most robust activation occurs with longer fasting durations, typically 3 to 4 days for humans. In summary, the minimum fasting length required to strongly trigger autophagy appears to differ markedly between rodents and humans due to differences in metabolism and physiology. In mice, a 48-hour fast is sufficient to induce significant autophagy and initiate a regenerative phase. In humans, achieving a comparable level of autophagy generally requires fasting for 3 to 4 days. This extended period allows for the depletion of nutrient stores, the switch to fat metabolism, and the upregulation of autophagic pathways across multiple tissues. The implications of these findings are profound. By harnessing the body's natural autophagic processes through carefully controlled fasting, it may be possible to promote cellular renewal, support immune function, and potentially delay the onset of age-related diseases. Ongoing research will continue to refine our understanding of the relationship between fasting, autophagy, and health, offering new avenues for enhancing human longevity and well-being. As our knowledge deepens, it becomes clear that autophagy is not merely a byproduct of starvation, but a fundamental biological process with far-reaching consequences. Through the strategic use of fasting, we can tap into this ancient mechanism, promoting resilience and renewal at the cellular level. The journey to unlock the full potential of autophagy is just beginning, and the discoveries yet to come hold promise for transforming our approach to health and aging. To further appreciate the nuances of autophagy, it is helpful to consider the molecular mechanisms underlying this process. At the heart of autophagy is the formation of double-membraned vesicles called autophagosomes, which engulf damaged organelles and proteins. These autophagosomes then fuse with lysosomes, where their contents are degraded and recycled. Key regulatory proteins, such as mTOR, mechanistic target of rapamycin, and AMPK, AMP-activated protein kinase, act as sensors of the cell's energy status, modulating the activity of autophagy in response to nutrient availability. During fasting, the inhibition of mTOR and activation of AMPK serve as critical triggers for autophagy. As energy becomes scarce, the cell downregulates growth and synthesis pathways, redirecting resources toward maintenance and repair. This shift not only preserves cellular function during periods of stress, but also helps to eliminate potentially harmful components that could contribute to aging and disease. The benefits of autophagy extend beyond simple cellular housekeeping. By clearing out misfolded proteins, damaged mitochondria, and other debris, autophagy protects against a range of pathological conditions, including neurodegenerative diseases, cancer, and metabolic disorders. In the context of aging, enhanced autophagy has been linked to increased lifespan and improved health span in multiple model organisms. The challenge lies in translating these findings into practical strategies for humans. While prolonged fasting can robustly induce autophagy, it is not feasible or safe for everyone. Researchers are actively investigating alternative methods to stimulate autophagy, such as pharmacological agents that mimic the effects of fasting or dietary interventions that modulate nutrient-sensing pathways. Nevertheless, fasting remains one of the most potent natural inducers of autophagy. For those who are able and willing to undertake extended fasts under appropriate supervision, the rewards may include enhanced cellular renewal, improved metabolic health, and reduced risk of age-related diseases. For others, shorter fasting periods or intermittent fasting may still offer meaningful benefits, even if the magnitude of autophagy induction is less pronounced. Ultimately, the decision to engage in fasting for autophagy should be guided by individual health goals, medical history, and personal preferences. Consulting with healthcare professionals and staying informed about the latest scientific developments will help ensure that fasting is practiced safely and effectively. The story of autophagy and fasting is one of balance between renewal and preservation, stress and adaptation, risk and reward. It is a testament to the body's remarkable capacity for self-maintenance and resilience, a capacity that can be harnessed through mindful engagement with our most fundamental biological rhythms. As research continues to shed light on the intricate dance between fasting and autophagy, we move closer to unlocking new strategies for promoting health and longevity. Whether through periodic fasting, dietary modifications, or future therapeutics, the promise of autophagy offers hope for a healthier, more vibrant future. In the end, the minimum fasting length required for autophagy is not just a question of hours or days, but a reflection of our evolving understanding of human biology. It is a journey that invites us to reconnect with ancient practices, embrace the wisdom of our bodies, and explore the frontiers of modern science in the pursuit of optimal health.